Please be seated. Welcome back. The next lecture is by old friend and Schwartz. Some of us did our postdoctoral training at Johns Hopkins University, and Schwartz is one of them. I'm another, a guy is another, so we are in a good company here. So Andy's post was with Georgopoulos, and Apostolos Georgopoulos. I believe that many of you in the audience know him, and since not all of you know him, he is the scientist who really made the major contribution to the technologies, to the understanding, to the technologies that we use today in brain-machine interfaces. Andy is from the University of Pittsburgh now, working together in different centers at the Pittsburgh University, like the Center for Basis of Cognition, Department of Bioengineering, the Garrett Institute of Regenerative Medicine, Department of Physical Medicine and Rehabilitation, and the Center for Neural Basis of Cognition. Okay, so he will tell us today his new view on motor cortical function, and I would like to invite you to give your speech.
So Elon and Guy are my old buddies from many years ago. As Elon said, we were at Hopkins together initially, and we've been in touch for now many years and had quite a few visits. So it's always really a pleasure to be here, because I think over the years we share a lot of same sort of scientific insight and philosophy, so it's always relaxing to come and have discussions here. So I just want to sort of emphasize that we have this emerging view of brain function that's quite a bit different than what we had 30 or 40 years ago. And that's the idea that there's a collective activity of the brain that's taking place continuously every minute that we're awake, and probably a lot of times when we're sleeping. That if you were to look at the brain en masse and be able to image the neural activity, you would see that most of the brain is active most of the time. And that is in stark contrast to this sort of classic way of thinking about brain function, where you think about the brain being composed of individual pieces and modules, each of which has its own private purpose. So that if you could split behavior into components, you would see the corresponding part of the brain light up for each of those behavioral components. And I think what we are really learning now is that that view is probably an old view and needs to be modified. So the idea is, if you just had a subject behave and draw a natural movement, use his arm to draw a movement, that you could then look at this activity and you would see something in this cartoon that looks much like this. You would see most of the brain firing, many of these neurons, billions of neurons firing in what looks like a fairly complicated pattern. And so as systems neurophysiologists, you could say our job is to look at that pattern and to try to understand something about what it means. And you could also decompose the problem to individual neurons. So we like to think of a neuron as having a specific job. Again, the classic way when we started 30 or 40 years ago, looking at individual neurons, we would record from this activity and then either do some manipulation, stimulate or ablate, or maybe even have the animal do a movement, and you would try to assign a job to that neuron, as if the only thing that neuron did was the thing that was related to your experimental paradigm. And so we got this, again, very narrow reductionist view of what these cortical neurons, or what these neurons were doing. And again, as time has gone on and we've looked at richer behaviors and applied more computational approaches, we realize that these neurons are really pluripotent. Not pluripotent like stem cells, but pluripotent in function. That these neurons do many things simultaneously. And that's an important idea, I think. So one way of thinking about this is you have inputs to these neurons, and you have to remember that just about every neuron in the brain has approximately 10,000 inputs, and it has about the same number of outputs. And there are about, different estimates of how many neurons are in the cortex, but there's something like 10 billion neurons. So you take 10 billion neurons with 10,000 inputs, it's something on the order of 10 to the 15th. And that's, when people talk about brain complexity, that's one way of looking at it. You have a connection matrix, something on the order of 10 to the 15th, which is, again, a very large number. But so if you look at this, and we go back to this old idea that, these neurons do individual things, they do specific things, and you then look at these 10,000 inputs and you say, does that make sense? And what we really would like to know is understand something about what's involved in these 10,000 inputs and how they make a neuron fire. In other words, if you could do the reverse and you looked at an action potential occurred that is a result of combined activity of all these synaptic inputs, and you said what was special about that combination of synaptic inputs that made that neuron fire, that would be a true understanding of basic neurophysiology. If you just ask a simple question, what makes a neuron fire? Really, we have no concept in a real behaving animal what that answer is at this point. So what we do instead as a surrogate is we just record the action potentials and then try to relate them to some aspect of our experiment. So what we do is, these action potentials generate an extracellular field. We introduce our electrode and record that extracellular field as action potentials. And the idea then is, if we understood something about the probability of seeing an action potential, could we infer what's making those action potentials? And so what was done, and again, as Helen said, in the early 80s was the observation from Georgopoulos that the direction of arm movement was important in making these neurons fire. And that was pretty radical at the time because, as you may or may not know, the idea of motor cortical physiology began as from an anatomical point of view. And so physiology consisted of animals that weren't able to move, they were paralyzed or decerebrate or asleep. And the idea is you would put electrical stimuli in one part of the brain and then look for a response in a different part with the idea that they were connected electrically. And so that was an anatomical idea. And then, in the early 70s and 60s, people were looking at things like locomotion. The Russians developed a decerebrate locomoting cat preparation, so you could actually look at movement and record neural activity at the same time. About in that same era, Evarts started looking at behaving monkeys that were awake, and he could record from cortical neurons. The movements were very simple. They were either elbow movements or wrist movements, single joint movements, because again, the idea was that what the motor cortex did was to activate individual muscles. And so you wanted to look at this muscle activity in relative isolation and try to relate that to neural activity. But shortly after that, as the experimental paradigms became more open, people and a couple Canadian investigators and Georgopoulos started to look at whole arm movements. So instead of constraining it to single joints and just worrying about muscles and joint displacements, he said, let's look at this whole arm movement. And when he did that, what he found was that he got a very simple relationship between movement direction and discharge rate. And so I'm going to illustrate that with this experiment. And what the animal did then was to just reach out in this virtual reality environment and draw with his arm. And what he was drawing was this template here. This is just a donut that looked like it was floating in three-dimensional space. Put his hand in this and drew around and around. And you can listen to a neuron that we recorded in the motor cortex as he did that. So here it is right here. And you could see this neuron's modulated very nicely as he moves his arm. And in particular, when he moves his arm upwards, the neuron fires, and when he moves it downwards, it stops. And so you don't need a fancy computer or a lot of analysis or a lot of terribly complicated equations to understand that there's this very robust relation between the direction of arm movement and cortical discharge. And it turns out that this relationship has been now found successively throughout almost the entire neural axis, from primary afferents to muscles to basal ganglia, superior colliculus, cerebellar nuclei, MT. There are a lot of places in the brain that have this kind of relationship to movement direction. And just to go back to what I mean by this relationship is that we sort of model this, and this goes back to the work of Georgopoulos again, and what he started out with was an animal that was using his whole arm to move a handle on a tabletop. So it was a planar surface, he had a handle, there'd be a target that would light up, the monkey would start from the middle of this table and move in these different directions, 8 different directions. And this was coined the center out task. I think everybody talks about that now, but I think it was really Georgopoulos that developed that task. And if you record a single neuron and look at the rasters that you get when you do this, so here the animal's moving to the right, and then here upwards, you see in this case, when the animal moved upwards, you see that the neuron fires a lot by this raster, and when you move downwards, the neuron shut off. And in between, there's a graded amount of activity. So if you were to plot movement direction along this x-axis, so again, our coordinate system is such that it's 0 degrees to the right, and then the mean discharge rate for each of these 8 movements on the y-axis, you would see this kind of relationship. So here's the mean and standard deviation across the 5 trials. You could see this, and what he did was to fit this with a cosine function. Now cosine functions, you say, okay, what should we think about the cosine function? I'm going to get into that, the inference about what a cosine function is in a minute, but just, grossly, cosine function is interesting because it spans the entire directional domain. So it covers all directions. It goes from 0 to 360. And it has a single peak. And that peak is the direction of that peak is what we call the cell's preferred direction. So it's a way that we label neurons. And it turns out that most of the neurons in motor cortex have this relationship. And they each have their individual preferred directions. And these preferred directions tend to be distributed throughout the directional domain. So let's just think about this for a second. So by fitting these data with a function, we're basically making a model. And our model looks like this. It's just a linear model. We say firing rate is related to the direction of movement. Movement has components, in this case 2 components, an x and y component. So that's the movement direction. And then what we have are these regressors, or you can think of these as slopes across a plane. And we fit this with multiple regression. We fit those B functions and get there and find them with multiple regression. So you can rewrite this equation in terms of vectors. So you can think that there's a vector describing the movement composed of these x and y components, and this B vector. This B vector has an x and y component. This B vector is the cell's preferred direction. So in the case of the cell I just showed you, this preferred direction was up. So you can think that this would probably be 1, y would be 1, and bx would be 0. Okay? Now, what does that mean? Well, it means that this equation is really a dot product. It's a projection of one vector onto another. And you can rewrite the dot product as the magnitude of vector b times the magnitude of vector m times the cosine of theta. This cosine of theta is that tuning function that I showed you before. So intuitively, what does this mean? Well, let's say you have a movement vector pointed up and to the right, and you have a preferred direction vector pointed this way. If you project this onto that, the length of this vector is proportional to the discharge rate of the neuron. So here, you see that when you move off and to the right, the neuron's firing at a little bit less than its maximum rate. If you move right, orthogonal to that, the vector gets very small, and really the neuron starts firing at its background firing rate. If you remember in that example I showed you when the animal's just holding his hand still, the neuron fires at a background rate. When you go opposite the preferred direction, the neuron actually goes below the background rate and almost shuts off. But the whole point is that the firing rate is proportional to the cosine of theta. Okay? And this is important. And the handy part about thinking about vectors is these vectors can be arbitrarily high dimension. So I showed you here a 2-dimensional vector. I could have a 100-dimensional vector, and the principle would be the same. You'd have some preferred direction in 100-dimensional space, and the discharge rate of the neuron would be proportional to how far your parameters in that 100-dimensional space were away from the cell's preferred direction. And that's important because I'm going to go through examples where I increase my dimensionality. Okay, so, you know, that's a way of thinking about what makes a neuron fire. But what we're really interested in is decoding. We want to look in the brain, and we want to do the opposite. We say we want to observe discharge rate and then predict the movement. Whoops, a lot of gestures here. So the way we do this is with a population vector. Remember, there's many cells firing simultaneously. They all have preferred directions. And so if I take that B vector I showed you and represent it by each blue line, one for each neuron, and then adjust the length of that blue vector by the observed firing rate, just like I showed you in that example, and I sum these together, and summing just means you take all the x components of the vectors plus all the y components of the vector, and you get this resultant vector, which we call the population vector, this yellow vector here. And what I'm going to do is calculate this every 30 milliseconds throughout a trial. So the animal's drawing, okay? I'll stop every 30 milliseconds, take the discharge rate off all the neurons, and build one of these population vectors. And I'll show you, if I hook these vectors tip to tail, that I can actually recreate the shape that the animal is drawing. So these are real data. The animal, we recorded from 360 cells, and the animal is drawing here, and you'll see what we were able to get. Oops, if I can do this right. So the animal is drawing a spiral from outside in, inside out. The spiral matched this template or this representation almost exactly. And so I can get the instant-by-instant representation, okay, of what the animal was really doing. And you can see this, again, it's isomorphic. It looks just like what the animal drew. It's accurate in time, and it is high fidelity. I didn't show you the actual, in this figure, I didn't show you the actual spiral, but it looks almost exactly like what he drew. Okay. Now, again, let me just back up. So this is back in the days when we could only record one neuron at a time. So what we did is we did this experiment over and over again. The animal was following a template, so the movements were relatively consistent. And then after we collected all the data, we created this population vector. Okay? Now, the other thing I want to say about the population vector is that population vectors do not really exist. Okay? They're just a computational tool. There's no node in the brain where this summation is occurring to create a population vector. All right, so what does this mean? Well, I think scientifically it means that we can actually decode natural movement, and we do this based on movement direction. And then the other thing to realize is that the coordinate system we're looking at is centered at the effector. So in this case, it was where the marker was that the animal was using to move the cursor, which was on the back of his hand, and that we get continuous coding. Okay, so we're not, selecting a template and letting it unroll ahead of time. We're actually calculating this movement on an instant-by-instant basis, or that's what it looks like the motor cortex is doing. Okay, the other thing, again, I really like to emphasize is that what we're pulling out of the brain is a trajectory. A trajectory, another question?
Yeah, the fact that the hand moves in a spiral way, has to be the computation takes place somewhere in the middle, not by a specific node in the brain, but the whole system. The computation does take place. Because of the
yeah, so I think what we're doing is looking at this tool. We are able to extract the information from the motor cortex. That doesn't necessarily mean that the motor cortex is the one that's doing it. Okay, it's just that we're able in that portion of the brain to extract the signal to be able to calculate this.
Do you think the summation does not take place anywhere in the brain?
I would say that there is no central node where this all comes together. the way I look at it is that the information stays distributed all the way through muscle contraction and joints until you make the movement. Okay, so it doesn't really come together until you make the movement itself. Okay, so what I showed you is that we're able to extract trajectories. You can think, you know, why is special about trajectories? Well, this really characterizes the way we move naturally, because what we're doing as we move is we're generating a time sequence of spatial points. This is, in my case right now, I'm speaking, so my tongue is moving a certain trajectory. That generates the sound that I'm able to use to communicate with you. Whether I'm walking around in the movement or gesturing with my hands, these are all trajectories. So it's really a way to relate what's going on inside your brain to the outside world. And the only way you can do this really is by moving. And you know, the other thing is we've done a lot of experiments with these drawing movements where we've shown that there's certain characteristics. Tamara flashes in here, but things like the 2/3 power law. All these characters that are characteristic of animate movement, these laws of movement, these psychophysical derivatives, are captured in this motor cortical signal. So what that means is that we can actually pull out movements that look like real movements. I think that's really important. And, you know, again, I apologize for those of you who have seen this. I like to use this quote from Moby Dick. So Melville was talking about Captain Ahab, and Ahab was pacing on the deck, and this guy was completely focused on this idea of the whale, the great white whale. And he writes, he said, if you watched Ahab, you'd fixedly gaze upon the ribbon-dented brow, you would see strange footprints, the footprints of his one unsleeping, ever-pacing thought. That thought would turn in him as he turned and pace in him as he paced, completely possessing him, that it seemed the inward mold of every outer movement. So again, what we're able to do is not just, you know, worry about muscle contractions and joint displacements and the mechanics of movement, but we're really being able to express what's going on in our brain. And so for people who are paralyzed and can't move, or, you know, people who are even locked in, or people who are, you know, paralyzed, they can't communicate fully with their environment, by extracting these signals from the brain and the fact that they're natural and animate and you know, are close to the real thing, we should be able to allow them to regain this lost ability. And so what we use is this electrode, and this is the Utah electrode array, originally developed by Dick Norman in Utah, and then really brought into widespread use with John Donoghue's group as they improved it over time. But what this is a cube of silicon. It starts out as a cube that's about 2 or 3 millimeters high. What they do is take a micro dicing saw and they dice it in 2 ways, and then they etch it. And after they etch it, they get these icicles, these posts right here, and with very sharp tips. They metallize the tips with platinum, and they insulate it. This is boron-doped silicon, so it's conductive. And then it goes down so that you can have nice recording tips, and then hook up to the backplane these wires I'll show you in a minute. So these are arranged as a 10 by 10 array on a 4 by 4 millimeter platform. Each electrode's 1.5 millimeters high. 96 of these are connected. And you turn this upside down and inject it into the brain. Here it is in motor cortex of a monkey. So here's the central sulcus. This array is in the proximal arm area, shoulder and elbow. This one's in the more distal area, wrist and fingers. Okay. And so, you know, the paradigm for neuroprosthetic control that, you know, that we use is something like this. You take the neural activity from each of the neurons that you're recording. So now you have the capability of recording about 200 neurons with these 2 arrays. You find each cell's preferred direction. That's indicated by this arrow. You can do something like a center out task to find the cell's preferred direction. You do this for all your cells. You add all those B vectors together to get the population vector. That results in this output right here that's the precursor to a trajectory or a velocity vector. This velocity vector then is fed to a robot controller, and the velocity that's being commanded is here at the endpoint of the effector. This tells the hand where to go. in this case in three-dimensional space, and we added another term to our vector, a fourth term that allows us to close the gripper. Okay? So the idea then is we can hold a piece of food out, the monkey can go for this, grasp it, and bring it to his mouth. So the model we're using, again, now we've expanded it. So the first model I showed you was 2 dimensions, these two. We added a third dimension so that the arm can reach in a volumetric space now. And then we added this fourth term for aperture. So we're up to 4 dimensions that we're controlling.
What's aperture?
Aperture is the hand opening and closing. It just opens and closes like that. So it's a continuous metric. So, you know, he can control how much he opens and closes. Okay, so here's the animal using the device. In this case, he decides he wants to lick the marshmallow off before he goes for the marshmallow. There's residue left on the hand. And he goes out and he can grab it just fine. Let's see. Here's one where it's more typical, where he just goes straight for the marshmallow. So this was done, you know, really in the period of 2005 to 2008. This was work that was done primarily by Mel Valiste, who was a postdoc in the lab. And you can see a monkey has exquisite control of the device. Okay.
A question?
Yes?
So in this case, the parameters for each neuron are fit during hand control?
No. So the way we fit this is the animal comes in the lab. He comes in the lab with his arms, both arms restrained. And what we do is we demonstrate this to the animal. Right? So we demonstrate the arm moving in different directions. And there's this phenomena called observation-based activity. So the neurons in the motor cortex respond in this situation in much the same way they would respond if the animal's moving his own hand. So simply by demonstrating the movement to the animal, we can get this center out driving and find the cell's preferred direction. This is a phenomena that's been observed by a lot of different laboratories, and you know, it's sort of a standard now, because imagine that the subject was paralyzed and we couldn't use the patient's own arm. This is the way we actually calibrate it.
And then you train?
Yeah, yeah, these are good questions. So this gives us a very rough estimate of the cell's preferred direction. And then what we have is we start the animal trying to do the task. He's not very good at it, but what we do is we limit the errors he makes. We call this ortho control for short. But what he does, when he moves correctly, the arm will move toward the target. If he moves at an angle away from it, we titrate the velocity so he can't move very much away from that to get him started. And then he does that, and then...
But you don't change the parameter.
Well, after he does that, he tries it, then we re-estimate the preferred directions again. Okay? So we go through a couple cycles of that. It takes about 10 or 15 minutes. And then once we've got that final set, we lock it into place. Okay? All right, so I showed you the animal reaching through 3D space and with a gripper. What we did more recently, this is work of Sam Clanton. What we did is we added a wrist to this effector. So we effectively now let the animal orient the hand, and we put a target cylinder out in space, and he has to reach out, orient the hand, and grab the cylinder. So these are the different orientations we use. And so our tuning equation now looks like this, where we have 3 for the displacement of the hand, 3 for the wrist, so your wrist can move in 3 different ways. And then we have the hand aperture, so now we're up to 7 degrees of freedom. And here's the animal doing that. So you can see he has to reach out and he has to orient the wrist to be able to grab this. Okay. So,
Can you check again how the parameters are related to the information that we
okay, so what I showed you basically was a tuning function that has 7 terms to it. So think of each vector as being 7-dimensional. Okay, so the population vector then is 7 dimensions. Okay, so I just showed you a 7-dimensional model. Now, we'd like to go to an actual hand. Okay? So the hand itself is incredibly complicated. So you can think that I could get away with 7 degrees of freedom, 3 for displacement, 3 for the wrist, okay, and then just simple hand. Now, if I want to increase the ability to use the hand, I have to look at a real hand. The real hand by itself has about 23 degrees of freedom. It's incredibly complicated, and it's a control problem. It's very tough. But fortunately, psychophysicists have looked at people as they grasp real objects, and they've looked at all the degrees of freedom, and what they find is that when you grasp, at least for simple grasping, that you have a set of correlations amongst these joints so that you can describe grasping as being composed of basic hand shapes. And then you mix these hand shapes together to get the real hand shape that you're using. So you have sort of these basis functions. And so what we've done then is we've used 4 basis functions in our tuning equation to get all the hand shapes that the animal would normally use. And so and the way we did this is by actually watching the animal move his own hand and record neural activity. So we had the animal reach out and grab a variety of objects, and we tracked the hand all 23 degrees of freedom very carefully with high resolution. And so we had him reach for a variety of objects that we presented to him robotically, things like push buttons and handles and pins that he had to pinch. And then what we did is we fit our neurons to now a 10-dimensional tuning function. So again, X, Y, Z space, a wrist, and now we have these 4 principal components describing hand shape. So now we're really ambitious, and our vectors are now 10-dimensional. And so we had the animal do this. Again, this was an animal that wasn't implanted with multi-electrode arrays, so we recorded 1 neuron at a time, had him do these stereotypic tasks, and then post hoc we recreated the movements, and this is what we got. So here's the actual hand shape of the monkey as he reached for those different objects, and here's what we're able to predict from that tuning equation. So you can see that, again, we're doing a very nice job. So the point was that, so what we did is we tracked the hand. Pardon me? Well, so what we did is we reconstructed these trajectories, and we took the average trajectory across all those. That's really what you're seeing, was the average actual trajectory. No, yeah, so what we're showing is, so we do this bin by bin, okay, across all the different hand shapes that he was doing. So what you're watching here, okay, this is constructed, this is the mean movement constructed across all those, okay, but normalized time across each trial. Okay, so at that time we didn't have a robotic effector where we could actually try to do this in real time, but luckily we were working with a defense agency, DARPA, in the US, and they had an ambitious program to develop a very high-performance prosthetic arm. They call this the MPL, Modular Prosthetic Limb. This is a project that's headed at Johns Hopkins Applied Physics Laboratory. This arm, total arm, has 17 active degrees of freedom, 26 total, because a lot of the finger joints are passive. The arm weighs about 9 pounds. It's the mean weight of a male adult arm. So it's made to be a prosthetic arm for amputees. It has this beautiful hand that has these fingers, each of which can move. They can ab and adduct. The thumb can be opposable, so it can actually move like that. It's really a very amazing device. It's a project that they've spent over $100 million developing. So here's what the hand looks like as a close-up. It looks pretty anthropomorphic. So with all that in place, we've done our homework, right? We did the basic science. We took each level of performance step by step in monkey experiments. We have now this high-performance arm. So we felt we were ready to start our human experiments So in 2012, we implanted our first patient. She was 53 years old. She's been paralyzed for 14 years. She has a kind of multiple sclerosis where she has a complete degeneration of her efferent tracts. She can't move anything below her neck. So here's for the neurosurgeons what we did. This was done at University of Pittsburgh. Elizabeth Tyler Cabrera was the neurosurgeon. So we exposed the cortex. Here's the dura. And this is the central sulcus here, so this is all motor cortex up here. And we implant these little arrays. Here you can see them being implanted. Again, there's one in the proximal arm area, one in the distal arm area. This is hand and wrist, shoulder and elbow. There was an extensive mapping study that was done with magnetoencephalography and fMRI, where the patient was imagining moving her own arm so we could pretty clearly delineate the area we wanted to go. Here you see the dura is now stitched back over, and you see these pedestals. These are the connectors that allow us to maintain contact with the recording arrays. Put the bone back, and we suture the scalp. So again, there's these two pedestals that remain exposed. Now, we were really excited when we did this, and she stayed 1 or 2 nights in the hospital, and we couldn't stay away from her. So we went into her hospital room, and we brought an amplifier, and we hooked it up to loudspeakers, and we just wanted to see what kind of responses we could get. So here it is. I think it's the day after surgery, and that was Elizabeth you saw off to the left there, the neurosurgeon. And I just have Jan. This is Jan, and I just say, imagine that you're going to move your arm, and then we're listening to this neuron fire. See, it's something you can't do with a monkey. So right when I said, think about moving your wrist, we could hear this neuron fire like crazy. So we were real happy because this was, right after the surgery. So a few days later, she came to the lab now, and now she's moving this modular prosthetic limb. And this was really the beginning of the second day of the experiment. She was able to move this in 3 dimensions right away. No extensive training. She was just imagining moving her own arm. For the first time in 14 years, she could move it. Move this robotic arm.
There you go. Yeah. Awesome. All right. See, the thing is the hand can go the other way. We can go in different directions. How about down here next?
So, this isn't, scripted by some canned program or anything. She's just, you can move anywhere you want. She can do it.
There we go.
Okay. So, this is, again, for those of us who are used to doing monkeys, this is really sort of enlightening and almost, it's like shackles are lifted from us. You can just think of experiments to do on the fly. So we had a news program coming and filming in the lab, 60 Minutes. And, we wanted to show some cool things that we could do. Can you hear that? Okay. So we just selected a neuron at random, or a nice looking one, put it up there and said, Jan, make that fire. So, but most importantly, what we did, question? Well, that's a great question, and we're actually working on that. So the other a lot of the other neurons are also modulated. So there are a lot of really cool experiments you can do with this sort of biofeedback, and we have a whole big program going on to describe that. So again, the most important thing that we want to do is show, does she have useful function? You know, can she use this really high performance arm to do something that's actually going to be beneficial to her? So there's a set of tasks that are called ARAT tasks that occupational physical therapists use to test patients that have had strokes. And they're a set of prescribed tasks, and they time them. So we selected that as sort of a way of doing that. And then we had her do this every day. So she came to the lab 3 days a week, and we had her practice this and do this over time. And so this is after 2 or 3 months of that. You can see the kind of things she's able to do. So this is, you can see Jan's eyeglasses right here. She's sitting in her wheelchair. And you know, we just put this block down and say, go for it, and put it on that shelf as fast as you can. And we time it to see how well she can do. And when you look at these movements, I want you to just look at that arm and see that, you know, you're seeing multi-jointed, coordinated movement that looks very natural. So here's another one. See, these are smooth, graceful movements. Now, granted, they're somewhat slower than, you know, a real natural movement, and we can talk about that later if you want. But you know, you get an idea of what she's capable of doing. This one I like because it demonstrates the full capability of, this is actually now you can see all 10 dimensions of that working simultaneously. So notice that she has to curl her little finger around to pick up this rock. And then she kind of splays the hand out as she tries to drop it here. So the idea then is that, sure.
Does it require mental effort from her?
Yeah, that's the interesting thing is that you know, we implanted an ECOG patient before, and the ECOG algorithm would work on sort of a spatial thing where one electrode would be for right and left movement, another, and that patient had a terribly difficult time to make this device work. On the other hand, with Jan, we have her just think about moving her own limb. And the more she does that without overthinking the task, and the faster she does it, the better she does. Okay? The problem we have with these subjects is that they're very competitive, and they don't want to make any mistakes. But one of the things that we found is we have to encourage them to just go for it, and if you have to fix it at the end of the movement. Don't worry about what's going on in between. And as she did that, it became completely intuitive for her to do. So the thing I wanted to show is that, you know, this isn't a one-off thing where we just sort of made a media event and, you know, demonstrated something for the, I will show you one of those in a second, by the way, you know, just, you know, to get a cool movie. We did this every day. And as we did this, this is now over 100 days, you see that her performance improved in a classic learning curve. And then, you know, if we look at, again, that ARAT task, how long it took her, there's a consistent decrease in the amount of time as she did this. She moved straighter over time, and she actually became more efficient in her movements. So this is a consistent trend that she was able to, you know, every day she was able to do this. Okay, so, you know, just a couple quick movies. So, you know, she could do whatever she wanted with this arm. And here, one day she decided, because we wanted to demonstrate hand shaping, she wanted to play rock, paper, scissors, and she wanted me to play with her. Trouble is, I never learned as a kid rock, paper, scissors, so she had to teach me. Okay, so, you know, what Jan really wanted to do was to eat a candy bar. So, for, you know, again, now this is something we didn't train on. So again, this was just a couple of days we tried this. So I will show you the first day when she picked up a piece of candy. So she picks up this candy bar. She's able to grab it right off the bat. But it's really the first time we allowed the arm to go anywhere near her face. And she's worried about smacking into her face, but she really wants to eat that candy bar. And you can kind of see how she's struggling with those two criteria. So we stopped the arm when it got to her face because we didn't want any bad things happening. But the next day, we just gave her full control. So notice, while she's chewing, she still has full control of the arm. Okay. All right, so that's kind of what I wanted to show you from Jan, and then I don't know, how much time do I have left?
2 minutes.
Okay, I'll skip a lot of stuff. So what we're going to do next in our next subject is we're going to implant, in addition to the 2 recording arrays, we're going to implant 2 electrode arrays in the sensory cortex and then activate those arrays electrically with the idea that we want to provide tactile and perhaps proprioceptive feedback back to the sensory cortex. And so these hands can be outfitted with these tactile receptors. There's also torque transducers inside there. You know, there's a variety of things we can put on this very elaborate hand to provide feedback. And so we're going to, again, give sensation back to the sensory cortex at the same time. Now, where can we go with this? I mean, you can think of what we'd really like to do is to use the subject's own arm instead of a robotic arm. And how might we do that? Well, there's ongoing work. Doug Weber, who's also at the University of Pittsburgh, has been implanting these Utah arrays in the dorsal ganglion of the spinal cord to record directly from primary afferents. And he's been very successful with that. He's been doing this in monkeys and has begun to do this interoperatively with a neurosurgeon in humans. So the idea then is we can get primary afferents from the subject's own skin and hands. We're just beginning a project with Scott Delp and Carl Disteroth to try to do optical activation of nerves to the muscles. And these will be actually in the nerve itself. But the idea is we can inject channel rhodopsin in these muscles. They get transported back to the spinal cord and alpha motor neurons. Alpha motor neurons make these channel rhodopsin proteins. They get inserted throughout the entire neuron, including the axon. So we can put light-emitting diodes, micro-light-emitting diodes over the nerve and activate these muscles. And you can imagine if we put different colors into different muscles that share the same nerve root, that from the same emitter we can activate these muscles individually. So that's the project we're working on at this point. So, and you can see this is work from Scott Delp, who's done this in rats. Now you can get nice recruitment curves when you activate these nerves with channel rhodopsin. So and here's some of the responses that Doug can get from his primary afferents. So the technology is right around the corner. And then shortly, one thing I want to say is that these electrodes, we've recently begun putting bioactive molecules on the electrode. So this is L1. This is actually a IgG-like surface molecule that's on neurons. This L1 is important in axon guidance during development. So Tracy Sui has put this on our electrodes. And here's a few days after implanting. This is, again, with a Utah electrode. This is just an example of the kind of recordings we can get. We actually were able to isolate 7 different beautiful waveforms on this one electrode, the largest of which, this one right here, was more than 1 millivolt, indicating that the neural element actually was on the surface of the electrode. And if you look over multiple days, typically from one electrode, you can get over 200 units. And these last, this is now day 75. We've gone out to hundreds of days now, and the number has gone down, but it's still way over 100 units. This is the mean amplitude of the units that you can get over time. So this is a very promising technology that we hope will allow us to do long-term recording of these electrodes. Thank you. Well, so John can answer this too, but I think John had a patient that was 8 years.
5 years and 4 months.
Okay, so you know the longest patient was that long. Typically, you know, I would say that the peak record. No. So what happens if you look at it? I think everybody's going to see this. You start out, and within about a month, you kind of reach the peak recording quality in terms of number of units and amplitude of units. And then there's this decline, and it reaches a plateau. And that plateau will last for years. So we're out with our patient now. She's about 20 months. And you know, she's about, I would say, half the number of units we had at the peak. But they're pretty stable. Okay? Yeah, so we actually have a whole series of experiments where we purposely manipulated the preferred directions. And the really interesting thing is the kind of learning you can do. So we can actually parse the learning out, because the only way the learning can be expressed is through those neurons that we record. So you can actually watch the way the neurons change their responses to correct for that perturbation. And there are two things that are going on. There's a very rapid response. It seems to be a change in strategy, so they're basically re-aiming at these imagined new targets. And then there's a longer-term thing where the preferred directions of the neurons are actually shifting. So we're able to actually parse those out in this kind of experiment.
How much excess information is there in the correlation between the units?
Okay, that's a great question. So what we don't really see, and this is pretty typical, you don't see this real short-term correlation very commonly between neurons, you know, on the millisecond or sub-millisecond scale. But longer-term correlation, you see this all over the place. And what you can then do is you can look at this and you can do dimensionality reduction on these neurons, and you can actually start pulling out common factors that are driving these neurons. And that's a whole nother arena of really interesting research. It turns out that these common factors, some of them are the prominent ones tend to be directional, but then there's some other ones that are very interesting that aren't directional.
What about extending beyond your own body? Having someone to control a quadcopter and bring something in the kitchen?
Yeah, so we're not doing that, but a lot of people doing EEG. I think there's Ben Heaty's group from Minnesota has this recent paper on controlling a helicopter with EEG. So yeah, people are working on that.
So maybe I missed it, but with the tactile feedback, are you trying to feed it back into the brain, or this is the decoder program that you have?
Yeah, so you're right. We could do it sort of just within the machine itself, but the idea is to actually activate sensory cortex to give the subject that information back. Now, whether it's going to be perceived as real touch, maybe not, probably not, but it still could be useful information in guiding the device.
Could you, for instance, translate it into a sound or something so the patient could hear it?
Yeah, we've actually done that already. So you can, adjust the torque sensors for gripping. So we actually have a tone that plays. So as she grips the object, she can judge how hard she's gripping it. No, we haven't used LFPs. I was going to show you, but we have done some state control using these principal components that are real nice. So it turns out that, in a nutshell, that you can easily distinguish when the animal isn't paying attention. When the monkey isn't paying attention, we call it idle state. You can see this as a discrete linear. If you either do LDA or principal component, you can see this as a component that lights up. So we know that we shouldn't be trying to decode during that period. It makes a huge difference.
All right, last question, yes.
So you said in time there was a plateau in performances over a few years or so. Is that caused because of the electrode kind of moving around, or?
So it's still an open question. There are a number of factors. There is indeed encapsulation around the electrode. So there's sort of scar tissue that builds up around the electrode. There's also a decreased neuronal density around the electrode. So there actually seems to be a dieback of neurons. There may be some, actually, they call it abiotic or material degradation. The electrode itself may not be as in great a shape over those number of years. So there are a number of factors, many of which we don't understand at this point. One interesting thing that seems to be a real take-home message is that if you make these electrodes small enough, so if you get the feature size down around 5 microns, that a lot of these degradative processes cease to happen. It's almost like if you're small enough, the brain doesn't see it.
Okay, I think we end here, and thank the speakers this morning again.